Assalamualaikum and hello everyone. My name is Nur Auni bin Kamsani. Today we are diving into the essential process of tissue growing in histology. Tissue growing involves the microscopic examination of specimen. This is essentially the first detailed look at the tissue samples, where all physical characteristics are recorded. It sets the foundation for accurate diagnosis by preparing specimen for microscopic analysis. In a histology lab, growing is carried out by trained professionals, including lab technicians, medical assistants, or pathologists. The person responsible for growing must examine, describe, and prepare each specimen carefully to ensure accuracy and consistency in later analysis. With the advancement in pathology lab, growing now goes beyond just handling specimen. It encompasses a workflow starting with specimen receipt, handling, and prioritization. Growing technology includes steps like specimen triage and sampling. Specimens are managed carefully from receipt through to embedding, with each step designed to maintain their integrity for later analysis. In the growing process, the knife and ink serve as tools of silent communication. This allows the growing technician to mark critical areas that need special attention, ensuring that histotechnologists and pathologists know exactly what to focus on. Certain features, such as the area of excision and mucosal part, must be identified. Noting these details ensure the specimen is embedded with the orientation that maximizes the diagnostic value. The conventional method of tissue growing involves the visual inspection, measurement, and manual dissection of tissue specimens to prepare them for microscopic examination. Growing typically includes steps like inking margins, taking measurements, describing textures or abnormalities, and preparing tissue samples in cassette for subsequent processing. Conventional growing is crucial for ensuring that relevant pathological features are captured and accurately documented. So, how tissue growing is performed? First, fresh tissue specimens come from various sources, so it is crucial to handle it with care since it can easily be damaged. The specimen needs to be placed in a liquid fixing agent immediately after being removed from the patient, such as formalin. Then, the specimen requires further dissection to select appropriate areas for examination. Care is always taken to prepare uniform, thin slices from large specimen with 3 to 4 mm maximum thickness. Lastly, place tissue samples into appropriate tissue cassette. Avoid overloading cassettes to allow reagent full access to each tissue sample. Clearly label cassette with patient information and specimen details to maintain accurate tracking. One of the primary challenges with this traditional method is time consumption. On average, it can take several minutes to manually grow each specimen, especially for a larger or more complex samples. These delays can ultimately slow down diagnosis, which is especially problematic for critical cases. Another major challenge is human error. In manual growing, even small mistakes like cutting the sample too thick or too thin can impact the quality of the slide, which may lead to the misinterpretation under the microscope. For instance, if the tissue is too thick, cellular details may not be visible, while tissue that's too thin might lack structural context. Next, contamination is another critical concern. If tools are not adequately cleaned between samples or if tissues are handled improperly, there is a risk of cross-contamination, which can produce misleading results. This is especially concerning in cases where accurate differentiation between samples is crucial, as contamination can lead to misdiagnosis. Another challenge is inconsistent documentation of grown specimen. This is due to traditional methods depend on written description and manual measurements, which can differ between people and often lack detailed visual. Written description lack of the capacity to fully capture a specimen's visual characteristics, such as texture, color variations, or subtle pattern. This inconsistency can lead to difficulties in case review, comparisons, and accurate diagnosis. So, what's the impact of this conventional method on diagnostic outcomes? When samples are mishandled or not processed correctly, the quality of the diagnostic information can suffer. Misaligned cuts, improper thickness, or contaminated samples may lead to unreliable type, risking a misdiagnosis or an inconclusive report. All these factors combine to contribute to longer turnaround time. Any delays in processing can impact patient care, especially in time-sensitive diagnoses like cancer. In oncology, timely diagnosis is critical for starting treatment as soon as possible. While conventional tools and methods have long been essential to histology, they do come with significant challenges that can impact diagnostic speed and accuracy. Addressing these issues is crucial to improve patient outcomes, particularly in critical diagnosis.
Assalamualaikum, I am Nur Amira Husna. Today I would like to present about advanced technique in tissue grossing process, which is macroimaging system. Macroimaging system facilitates the efficient capture, annotation, measurement and storage of high resolution image of gross specimen. As we can see within the picture over here, the system consists of few crucial components in order to facilitate greater diagnostic accuracy. The next one is principle. The system consists of few components, which is the first one is a closed HD camera to display live video at 30 frames per second at HD resolution. The camera also can be controlled hands-free with the foot pedal. Furthermore, the camera is roughly the size of a coffee cup, thus requiring very minimal workspace. The system also consists of a touchscreen monitor. This monitor is designed to be handled with a glove finger. This allows users to seamlessly interact with the monitor without removing their glove, thus reducing interruptions and enhancing workflow continuity. Annotations can be directly added on the screen to mark specific areas of interest or labeling structure for reference. Furthermore, barcode scanners which help to identify and track cases and also to guarantee the quality assurance. Barcode scanners accurately tracking and linking specimens to their corresponding data to reduce the risk of errors and ensuring that each sample is properly identified throughout the workflow. Lastly, this macroimaging system consists of imaging software. Images captured are connected to software that enables live streaming from the grossing room at a resolution of 1080 pixels, thus providing highly detailed pathology consultations to enhance diagnostic accuracy. Furthermore, the software only consists of a simple icon with one-click access to essential control, thus make this system a pleasure to be used. Moving on to the next part, which is the effect of this macroimaging system to the diagnostic process. The first one is specimen can be virtually sectioned for pre-assessment. This pre-assessment helps in identifying key regions of the interest to optimize tissue sampling. Next one is enhanced documentation and record keeping. This is due to macroimaging produce precise digital records that can be efficiently stored, referenced, and shared. This is crucial for quality control and comparative analysis of a specimen. Benefit of macroimaging system. The first one is enhanced visualization to facilitate accurate tissue examination. This is due to the ability of this technique to provide clear, accurate visuals of a tissue, making it easier to analyze and interpret anatomical details. Next one is optimized workflow efficiency by reducing time spent on documentation. This technique can capture and label tissues in one step, thus reducing time for manual documentation. Lastly, improve communication between pathologists. High-quality image enhance communication between the grossing team and pathologists by precisely illustrating specimen features such as margins, orientation, and abnormalities. As a result, unified understanding across all parties can be achieved.
The next advanced techniques that have been used in tissue grossing is related with tissue marking and orientation aids, which can be divided into two. The first one is specimen inking with fluorescence dyes, and the second one is laser marking. Traditionally, color ink have been used to mark tissue margins, but now advanced methods like fluorescent dyes and laser markings are making grossing even faster, more precise, and less prone to error. These techniques fit seamlessly into the grossing workflow. Fluorescent dyes and laser marking allow laboratorians to quickly and accurately mark critical errors on the tissue. This means less time spent on each sample and a more organized process, especially in high-volume labs. So how does this technology work at the grossing stage? So here's the principle. With fluorescent dyes, tissue samples can be marked with color that glows under specific wavelength of light. This helps identify critical areas quickly and clearly to do the grossing. Even after further staining, these markings remain easy to see, making it simple for pathologists to understand the tissue orientation. Then, there's laser marking. This technique uses high-energy light to precisely mark tissue surfaces without adding chemicals that might affect the sample. Laser marking are used to enhance orientation, ensure correct alignment during the sectioning, and maintain the sample quality for later tests. For tissue grossing, these techniques make a big impact on the diagnostic process. Fluorescent dyes and laser markings allow laboratorians to make tissue faster and more accurately, ensuring samples are ready for analysis with clear orientation. This precision leads to faster, more reliable diagnosis, benefiting both the lab team and the patient. At the grossing stage, these new techniques offer several key benefits, which are: the first one is greater precision. This helps laboratories to clearly identify important tissue areas with the help of high contrast markings. Second, improve workflow efficiency by reducing the time spent handling each sample and reduce manual handling errors. The third one is improve digital compatibility, where laboratories are able to work seamlessly with digital imaging through fluorescent dyes. And last but not least, preserve sample quality. This is because there's no added chemicals to interfere with tissue analysis. In the end, these advanced histological techniques in tissue grossing make grossing faster and more precise, setting a strong foundation for accurate diagnosis compared to traditional methods in tissue grossing.